Welcome to the BioTech Whisperer channel. Our topic today is on a perspective overview of CRISPR gene editing. If you are new here, welcome you to join us where we as a group of retired professors sharing bite-sized videos in life sciences. Let's dive into the topic for today. Genome editing, also known as gene editing, is a set of technologies that enable scientists to alter an organism's DNA by adding, removing, or modifying specific locations in the genome. There are several different approaches to genome editing that have been developed. CRISPR-Cas systems, including CRISPR-Cas9, are part of the immune system of prokaryotes, single-cell organisms without a nucleus, and protect them from viral infections by cutting the nucleic acids of invading viruses. CRISPR-Cas systems are part of the adaptive immune response of prokaryotes, such as bacteria and archaea. When a virus infects a prokaryote, the CRISPR, clustered regularly interspaced short palindromic repeats, sequences in its genome are transcribed into short CRISPR RNA, crRNA, which guides a CRISPR-associated Cas protein to cut the viral DNA or RNA. This prevents the virus from infecting the prokaryote again. Initially, CRISPRs were thought to be a DNA repair mechanism in thermophilic archaea and bacteria, but in the early 2000s, researchers discovered that the spacer sequences between the DNA repeats in CRISPRs were similar to sequences found in viruses and plasmids. This led them to realize that CRISPRs play a role in the adaptive immune system of prokaryotes. Timeline. CRISPRs. They were first discovered in 1987 by a Japanese scientist, Yoshizumi Ishino, and his team, who were studying a gene responsible for converting alkaline phosphatase. However, the function of these sequences was not understood at the time. In 1993, researchers led by J.D. van Emden in the Netherlands found that different strains of Mycobacterium tuberculosis had different spacer sequences between the DNA repeats. They used these spacer sequences, known as polytyping, to characterize tuberculosis strains. Subsequently, CRISPRs were found in several other bacterial and archaeal genomes. Researchers Francisco Mojica and Ruud Janssen were the first to refer to these sequences as CRISPRs. In 2012, George Church, Jennifer Doudna, Emmanuel Charpentier, and Feng Zhang developed a way to use CRISPR-Cas9 as a tool for editing specific regions of genomes, which has the potential to revolutionize gene editing. Because of its significance, Science magazine named CRISPR the Breakthrough of the Year in 2015. CRISPR-Cas systems have several components in addition to CRISPR, sequences and Cas, CRISPR-associated proteins. Cas proteins are enzymes that cut DNA or RNA at specific sites, and their role as nucleases was discovered through comparative genomic analysis of CRISPR and Cas genes. Some Cas proteins, like Cas9, cut DNA, while others, like Cas13, cut RNA. Another important component of CRISPR-Cas systems is the protospacer adjacent motif, PAM, a short sequence of 2 to 6 base pairs in the viral genome that is located next to the sequence targeted by the Cas nuclease. PAMs are essential for the function of CRISPR-Cas systems because the Cas nuclease cannot cut DNA without a PAM present. PAMs ensure that only foreign viral nucleic acids are cut, while CRISPR arrays are left intact. CRISPR-Cas9, CRISPR-associated protein 9, system could be used as a tool for genome editing by designing guide RNA to target a specific region of the genome. This allows the CRISPR-Cas9 system to be used as a cut-and-paste tool to modify genes by removing, introducing, silencing, or activating them. CRISPR-Cas9 has been used to increase the production of lipids in microalgae for the production of biofuel and has the potential to be used, in the future, to cure genetic disorders such as sickle cell anemia and cystic fibrosis. There are also a wide range of applications for CRISPR-Cas9 in the treatment of cancer and infectious diseases. Genome editing has the potential to be used in the prevention and treatment of human diseases. It is currently being studied in cells and animal models, in research labs, to understand diseases and determine whether it is safe and effective for use in humans. Research and clinical trials are exploring the use of genome editing for a variety of diseases, including single-gene disorders like cystic fibrosis, hemophilia, and sickle cell disease, as well as more complex conditions like cancer, heart disease, mental illness, and HIV infection. Since the discovery of the CRISPR-Cas9 gene editing system, researchers have modified the Cas9 nuclease to perform targeted epigenome editing. This modified Cas9, called enzymatically date Cas9, dCas9, can be linked to enzymes that modify the epigenome, such as DNA demethylases, methylases, or acetyltransferases. Like unmodified Cas9, dCas9 is directed to a specific region of the genome using a guide RNA, but instead of cutting the DNA, the dCas9 enzyme complex modifies the epigenome at that site. Epigenome editing can activate or suppress gene transcription, depending on the specific enzyme used. While it is still early in the development of CRISPR-Cas9 technology, the potential applications are vast. In our next video, we will review the current understanding of Sherlock CRISPR technology. There are ethical concerns surrounding the use of genome editing technologies, such as CRISPR-Cas9, to alter human genomes. Most of the changes introduced through genome editing are limited to somatic cells, which are cells other than egg and sperm cells, germline cells. These changes do not affect future generations because they are not passed on to offspring. However, changes made to germline cells or to the genes of an embryo could be passed down to future generations. This raises ethical issues, such as whether it would be acceptable to use this technology to enhance normal human traits, such as intelligence or physical abilities. Due to these ethical and safety concerns, germline cell and embryo genome editing are currently illegal in the United States and many other countries. Building on the CRISPR gene editing system, MIT researchers have designed a new tool that can sniff out faulty genes and replace them with new ones, in a safer and more efficient way. Using this system, the researchers showed that they could deliver genes as long as 36,000 DNA base pairs to several types of human cells, as well as to liver cells in mice. The new technique, known as PASTE, could hold promise for treating diseases that are caused by defective genes, with a large number of mutations, such as cystic fibrosis. Amongst other technological innovations, we have Mini CRISPR-Cas12F1 as a new genome editing tool. The CRISPR revolution has just begun and we expect more to come. Stay tuned to BioTech Whisperer channel. Stay blessed.